A very good evening to everyone. Welcome in today's class and let's crack GATE together. So this is the channel Let's Crack CSIR UGC NET and we are doing the crash course for GATE 2021 every day evening till the GATE exam comes. Right? So every day 5 p.m. we'll be preparing for the GATE examination. I'll be dealing with biotechnology and the topic of today is artificial seeds. So, Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET and GATE examination, and we are reading biotechnology paper, right? A little introduction about myself. I'm your educator Farida Bandookwala. I have a teaching experience of 10 years in the same field. I've qualified MP SLET examination and presently I'm pursuing my PhD from School of Biotechnology, DAVV, Indore. Before beginning with the topic that is artificial seeds, let us have a small introduction about Unacademy. If you are opting Unacademy, what all things you are going to get? You are going to get daily life classes so that you can be motivated and you can always be in that motion of learning. And from today, we are starting the crash course for GATE 2021. So we'll be doing daily life classes from Monday to Friday, and on Saturday and Sunday also we'll be having some kind of sessions. The syllabus is in well structured manner because you have to complete a lot many things in a small span of time. So the syllabus should be very much structured. The course must be very much structured so that you can make the most out of it. Then we'll be taking live tests and quizzes so that you can evaluate yourself that how much you have learned so far, which is quite necessary for qualifying any kind of competitive examination. Then you will get unlimited access towards all the videos which is available on the platform. These free live classes Unacademy takes on YouTube and it takes on a special platform. So presently we are doing a class on YouTube. So this is a free live class. You can just subscribe our channel and get the benefit of free live classes. How you're going to get special classes? You need to download the Unacademy Learning app. When you will download this app, you need to make your account. You need to log in and then you can search by my name, that is Farida Bandookwala, and you will come across my profile, follow my profile, and like that you will get the access towards all the special classes which I have conducted previously and all the upcoming special classes which I'll be taking. If you are subscribing for Unacademy, then you will get the access towards all the educators, not only me but all the educators. Then there is this plus subscription. So by this plus subscription, you will get the access towards all the plus classes which are done by any of the educator in the same field, that is CSIR or GATE. Another thing about this is it's a paid subscription, but the amount is very pocket friendly because presently a major discount is going on, that is 54%. Just opt for a yearly or 2 yearly program and you will be able to make the most out of this videos and things available on the platform. We provide the notes in the form of PDFs. We conduct tests and quizzes. We conduct live classes so that you can ask your doubts, queries, and everything can be crystal clear. So just go for a yearly or 2 yearly program and make the most out of it. After that, if you're using my referral code, that is FB01, you'll be getting an additional 10% discount. So do not forget to use my referral code, that is FB01, and avail additional discount of 10%. If you're going for 12 months, the course is going to be very cheap, that is 10,395, and you're opting for 24 months, the course is going to cost you 15,120 rupees. It is very pocket friendly, and recently a discount is going on and soon the prices are going to hike. So get the subscription as soon as possible and make the most out of this time, because presently I think no offline classes are going on anywhere in India because of this pandemic. So just make the most out of it. So today is the day to get the subscription. Don't wait for tomorrow, okay, I'll go for tomorrow, because very less time is left. Take your subscription now, today itself. New batches are starting on plus subscription. Also new classes are starting from today, so the time when you are going for this subscription is now. just take this subscription and start your learning. Now coming to the topic, that is artificial seeds. That is the topic, right? Artificial seeds. This is the topic, right? Now, if you want to understand artificial seeds, you need to know about somatic embryogenesis. But as we are learning artificial seeds, so let us talk about artificial seeds a little, and then I need to tell you the concept of somatic embryogenesis. So, what are artificial seeds? Artificial seeds are living seeds like a structure, but they are made experimentally by a technique where somatic embryos are derived from plant tissue culture and they are encapsulated. Right, so I've given you a lengthy definition, but let us write this, that what are artificial seeds? They are living seed like structures which are made experimentally by a technique where where somatic embryoids derived from plant tissue culture are encapsulated by a hydrogel and can be used as true seeds. This is the definition of artificial seed. Recently, some kids are playing in my neighborhood and I think there is lots of disturbance. So please ignore this disturbance because some kids are making lots of noise. From tomorrow, I'll try to opt for some another place where I can take the classes in a well quiet place. So sorry for this. Those who are attending the class live, they can write in the comment box if they are like really disturbed by the sound. Coming back to the topic, that is artificial seeds. They are actually living seed like structure. They are not actual seeds, but they are living seed like structures, and they are actually made experimentally. They are not made in nature, they are made experimentally by a technique, that is the somatic embryoids, which are derived from plant tissue cultures, and they are then encapsulated. They are captured in a hydrogel, and then they can act as true seeds. So this is the definition of artificial seeds. Now after this we need to talk about somatic embryos. So somatic embryos or somatic embryogenesis, that is the actual topic which we should study before the artificial seeds, because this is the thing which is the basic of artificial seeds. So let us start with this, that is somatic embryogenesis. Somatic embryogenesis. So the process of fertilization triggers the formation of an embryo, and this mechanism is called as embryogenesis, right? Fertilization and then embryogenesis. But what if fertilization doesn't take place? So is it possible that without fertilization embryogenesis can occur? Is it there in nature that naturally some kind of embryogenesis occurs without fertilization? So who is going to answer me in the comment box? Yes, there is an answer to this, that is parthenogenesis, right? This is the thing through which there is no need of any kind of fertilization. Directly the somatic cells can get converted into embryos. And this parthenogenesis, it doesn't occur in higher plants, but it occurs in lower plants, isn't it? Right, we know this. So parthenogenesis is defined as growth and development of embryo without fertilization of the female gamete. Right, parthenogenesis occurs in lower plants, no need of fertilization. No need of fertilization. So we know this. So in nature has shown that yes, it is possible in case of plants that without fertilization something can occur. So in nature, it is observed that tissue that are part of embryo sacs or those that surround it can also develop into an embryo, right? So, however, particularly environmental condition which exist inside the embryo that will be required for this process. And from here, the idea of somatic embryogenesis was derived. So now I'm going to tell you what is somatic embryogenesis exactly. Now we know the introduction, at least we know something about it, that yes, it occurs in nature and how this idea came into the mind. So what is somatic embryogenesis? It is defined as development of an embryo derived from somatic cells, right? No germinal cells are needed. So somatic cells which are used for formation of embryo is somatic embryogenesis. But under proper environmental condition, proper artificial in vitro condition, that is the prerequisite. Otherwise, it cannot occur. So it is a very significant process in plant tissue culture and it offers a rapid large scale propagation system that is useful for many industries, right? So what is somatic embryogenesis? Development of an embryo derived from somatic cells under appropriate artificial environmental condition. This is the literal definition of somatic embryogenesis, which is the most important thing in artificial seeds, because in syllabus they have written artificial seeds, but artificial seeds, they come from the process that is somatic embryogenesis. So somatic embryos, they develop and differentiate in the very same way like zygotic embryos. There is no difference in that. So let us write this sentence also because it is quite important. You should know this one liner, that is somatic embryos develop and differentiate the same way as zygotic embryos, and they also pass through the similar stages. So what are the similar stages? Pass through similar stages. So that is proembryo, then globular, then torpedo, and lastly cotyledonary stage. Cotyledonary stage. These are the 4 stages through which our somatic embryo it passes. Right. So the other terms which are used for the embryos which are developed by this process, they are. So we need to know the synonyms also for the somatic embryogenesis or the embryos which are developed from this, right? So what are the other terms used for the embryos which are developed through this process? That are adventitious embryos, accessory embryos, then embryoids or embryo-like structure. These are some of the terms which are used for somatic embryos, right? These 4 terms: adventitious embryos, accessory embryos, embryoids, embryo-like structures, right? These are the things. Now they we have seen that it passes through following stages. So there are developmental stages of somatic embryo. So we will see that what are the developmental stages. So developmental stages of somatic embryos, of somatic embryos, right? So what are these? There is one cell stage, then there is two-celled stage, then there is four-celled stage, then there is globular stage, globular stage. So we can draw the diagrams also for this because they have a very peculiar structure in them. So when we are saying one-celled, two-celled and four-cell stage, so it will be like this, and then when we are saying globular, so it will be like this, it will be kind of a globule. This is globular stage. Then after that, heart shaped stage, heart shaped stage. So how does this heart shaped stage would look like? Somewhat like this. So it will be like completely filled with cells. After heart shaped stage, torpedo stage embryo. So how this torpedo will actually look like? It will be like elongated heart. This is like torpedo stage. So one-celled stage, two-celled stage, four-celled stage, then globular stage, then heart-shaped stage, and then torpedo stage embryo. And after this, after torpedo, there will be well-formed plantillets. Well-formed plantillets. So plantillets. So it will have shoot as well as root. Shoot plus root, right? So first stage, second stage, third stage, fourth stage and fifth stage. So remember globular, then heart, then torpedo and then plantillet, because this sequence can be asked in examination, right? That which is the choose amongst the following, which is the sequential stage for the development of somatic embryos. So we'll be going from one cell, two cell, four cell, then globular, then heart, then torpedo, and then well-formed plantillets. We know the sequential questions like which are asked in evolution, which man came first and which man came last, like those all Neanderthals, Homo habilis and all those things. So that type of question can occur for somatic embryos as well, right? Now somatic embryos can be of two types of somatic embryos or somatic embryogenesis. So first is primary somatic embryogenesis, primary somatic embryogenesis, and second is secondary somatic embryogenesis, but obvious the term is quite okay. So primary somatic embryogenesis, which thing will be called as primary somatic embryogenesis? In this type, the embryogenesis can only be induced by using the explants, right? I hope you people know the concept of explants. Those who are attending the class live, they can just write in the comment box, do they have the idea what is this explant? And if they don't have any kind of ideas, we'll be discussing it, right? What is somatic embryogenesis? So in this phenomenon, the development of embryo is under the induced through existing somatic embryos. So this primary somatic embryogenesis will occur directly from explant, and in secondary it will occur through existing somatic embryos. So I haven't got any answer. So let us discuss explants. No worries. What is an explant? What is an explant, right? So explant, whenever we are opting for a part of the plant, like for example, this is a leaf, and we are going for the tip of the leaf because it has got lots of meristematic tissue. So usually we go for that, or tip of the plant, or newly budding leaf. These all things can serve as explant. So there's a small piece of leaf which will be now used for development of embryo artificially in plant tissue culture. It is called as explant. So Yeah, I should have told you this initially, that somatic embryogenesis is a part of plant tissue culture. This whole thing comes under plant tissue culture, right? So explant is that small part of the plant which is used for development of whole plant or somatic embryo or whatever thing we are calling it. So that is an explant, right? So this we know now. What is an explant? And primary somatic embryogenesis is what? If we are going with this small part of explant and we are keeping it in the media, and directly we are inducing it for developing into somatic embryo. It is called as primary somatic embryogenesis. And if we have developed this explant into a callus, what is a callus? What is a callus? Callus is undifferentiated mass of cells. Callus will be slightly yellowish colored. It will be undifferentiated cell. It is having the potential to develop into a whole plant, into only roots or into shoots, anything. This is our callus in plant tissue culture. So if we are using existing somatic embryos to develop into the whole plant, it will be secondary somatic embryogenesis, right? So directly if you are using explant for changing into embryo, it is primary somatic embryogenesis. And if you are using existing somatic embryos for developing into somatic embryos, it will be secondary somatic embryogenesis, right? So these are two types of somatic embryogenesis. Now there are again two ways to induce the somatic embryogenesis, right? So the two ways of inducing somatic embryogenesis. What are these two ways of inducing somatic embryogenesis, right? So first is direct somatic embryogenesis, and second is indirect, right? So what is direct somatic embryogenesis and what is indirect somatic embryogenesis? So in direct somatic embryogenesis, the embryo is developed without any intermediate callus stage. The embryo can be developed directly inducing the explant for the genesis. So that is our primary somatic embryogenesis, direct somatic embryogenesis. And in indirect somatic embryogenesis, the development of an embryo occurs with the help of intermediary callus. So we'll write occurs through intermediary callus. So first of all, this explant will develop. This is explant. This explant will change into callus, and this callus will change into somatic embryo. This is indirect somatic embryogenesis, and in direct, this explant will be kept in medium and directly it will be changed into somatic embryo. So this is direct somatic embryogenesis. Right, I hope I have made this point clear. Now, when we are reading artificial seeds and we are reading somatic embryogenesis, we should know the application of somatic embryos, and then we'll be moving towards the artificial seeds. So, application of somatic embryogenesis. So, first application, it is a preferred in vitro propagation method for the woody plants. So, it plays a very, very crucial role in clonal propagation. Like if you are just taking a small explant and you are developing it into callus, and then you are cutting callus into small pieces, and those small pieces, maybe 100 pieces, now you are using for development of 100 plants. So, isn't it a very easy method? So it plays a very crucial role in clonal propagation, because all the plants, like for example a mango tree which is giving a very pulpy mango with a very nice flavor and it is high in demand in market, and if you'll go for the conventional reproduction in that mango tree, there will be some changes, but no, people are loving that mango tree. So what we will need to do, we need to make many clones of this mango tree so that the demand is fulfilled. So how is it possible? That is possible through somatic embryogenesis. That is a part of clonal propagation. And if we have to supply this plant to some other place, that its demand is in some Arab country or its demand is in European country, so we can make artificial synthetic seeds, or we can make artificial synthetic seeds and give it to farmers. We can transport it. So That is its application. Then germplasm conservation, then cryopreservation. These all are the application of somatic embryogenesis. So we'll write this, that it is helpful in vitro propagation of woody plants, of woody plants. Then it is highly helpful in clonal propagation. Then it is useful for synthetic seed production. Then fourth, it is used for germplasm conservation. So obviously, if we are making somatic embryos and we are storing it, and we can use it after some years, maybe 10 years, 15 years, 20 years, 100 years. So that is a kind of germplasm conservation, right? Then it is used for rapid large scale propagation of plants for the production of secondary compounds and drugs. There are many plants which are used for their secondary drugs or secondary compounds. So that is also possible with the application of somatic embryogenesis. So secondary compounds and drugs. Secondary compounds and drugs. After that, it is an ideal system for the basic studies of plant cell biology, embryo development, like we have seen the stages through which it crosses. So those all things can also be seen very nicely through somatic embryogenesis in vitro condition, right? And it provides a very good system for understanding the differentiation and mechanism of totipotency expression in plant cells. So sixth, we will see various study purposes. Various study purposes. So differentiation, totipotency, various experiments of cell biology and embryo development, these all things are possible through somatic embryogenesis, right? So now we know a very good amount of somatic embryogenesis, so we are well equipped. Now we can move into the second part, that is artificial seed. Though I have given the topic artificial seeds, but it cannot go without somatic embryogenesis, right? So artificial seeds. Now we come to the actual thing. This is written in the syllabus of GATE. It is written artificial seeds, but without somatic embryogenesis you cannot move ahead, right? So let us talk about artificial seeds. So what are artificial seeds? We have already seen the definition, isn't it? Let us go back to the first page. So what are artificial seeds? They are the living seed-like structure. They are living, definitely, right? They are living seeds-like structure which are made experimentally. They are not developed by nature, but they are made experimentally by a technique which is called as somatic embryogenesis. And then after that, they are packed into some kind of structure. If we are going for, let me take the new page and I'll show you. So yeah, so imagine that this is a callus, right? This is a callus. So callus is undifferentiated mass of cells, right? So if this callus is used directly or given to farmers, so is it possible that they'll be able to handle it? Because anything which is in a very primary stage is very vulnerable because of the environment, because of the handling. Everyone cannot handle everything, right? So this callus, it is very fragile, very vulnerable thing. So it needs to be changed into a sturdy thing so that it can be transported from one place to another, and it can be even given in the hands of layman, right? So that is the place. So that is the place where we come across the concept of artificial seeds. That is, they are encapsulated. What I was trying to tell you, that encapsulation is necessary. Now we are done with somatic embryos. We have learned that how somatic embryo is developed, right? Through plant tissue culture, we need to have an explant, then we need to go for direct embryogenesis or indirect embryogenesis or primary somatic embryogenesis or secondary embryogenesis, right? We are well equipped with this thing. Now we have this fragile embryo with us. Now we cannot give it like that. We need to pack it in some sturdy thing and then we have to distribute. So that is the place where artificial seeds come into the being. So now we know the definition, we have seen the definition, isn't it? Now we need to go for the method of artificial seeds. We have seen the definition in the very starting page. So now we will be talking about the method. So first step will be establishment of callus culture, establishment of callus culture. Those students who are attending the class live, please they can write me in the comment box if they are facing any kind of disturbance in terms of audio. So establishment of callus culture, then induction of somatic embryogenesis, in callus culture. Now you people know what is a callus, what is a explant. So do not get confused at all. After that, maturation of somatic embryos, maturation of somatic embryos. After that, encapsulation of somatic embryos, encapsulation of somatic embryos. So after encapsulation, what is encapsulation? Like imagine if this is a somatic embryo and we make a covering of something. So this is the capsule. This is the capsule and this is the somatic embryo. So this is encapsulated somatic embryo. Now after this, after encapsulation, the artificial seeds, they'll be tested. They'll be tested for their viability. So how will be they tested? Encapsulated somatic seeds are tested, right? Whether they are capable of developing into a complete embryo or not. They are tested for embryoid, plant conservation, plant conversion, and greenhouse field planting. Greenhouse field planting. These are the two ways they are actually seen, whether they can actually change into plantlet or whether they can actually survive in greenhouse or they cannot survive. So these are the things through which artificial seeds, they are tested. Now we know the steps till here. Now we have to talk about encapsulation process, right? We haven't talked about this till here. It was part of somatic embryogenesis, so we know it well, we have learned it. Now encapsulation, this is the thing we need to talk about. So I'm going to tell you four methods, the four very famous and quite being used in long term, and from very long term it has been used. So for those four things I'm going to tell you. So there will be a gel for this thing and there will be a complexing agent. When we'll be taking gel, we need to make the complex of gel so that they become hard. So there will be a gel and there will be a complexing agent. These two things together will make our artificial seeds capsule. So first is sodium alginate. This is the most famous. In fact, those experiments which students they perform at lab level in university and colleges, they use sodium alginate, and the complexing agent that they use is calcium salts. Right? I'm not going with the concentration because that is not much required over here, because we are not doing the experiment. We just need to know if you are using sodium alginate, the complexing agent would be calcium salt. After that, sodium alginate again, sodium alginate plus gelatin. So the complexing agent in this case will be calcium chloride. After that, the third thing, that is carrageen or carrageenan plus locust bean gum. And the complexing agent in this case will be potassium or ammonium chloride. The fourth gel will be Gelride. This is a company product, Gelride. And when does it become complex? So it is a temperature dependent. So whenever the temperature is lowered, it changes into the complex. So these are the four examples of the gels and the complexing agent which we should use for making the encapsulated synthetic seeds or encapsulated somatic embryos. One and the same thing. If we are saying encapsulated somatic embryos, it means artificial seeds, right? And we are not talking about the hormones, that is gibberellins or cytokinins or anything, because that is altogether a different matter. We just need to know what is artificial seeds. Now there are two standard methods which are used for coating the somatic embryos. We know that these four things can be used, but now the way they will be used, that is also one thing. So the two ways through which it can be used, those two things we are going to discuss, right? So coming to those two things, two standard methods, two standard methods for coating somatic embryos, for coating somatic embryos, right? So first method is gel complexion, gel complexion via a dropping procedure, via a dropping procedure. I'll tell you both of the things. And second is molding. These two are the ideally used method for making the artificial seeds. So if we are saying gel complexion via dropping procedure, so what we'll do, so the isolated somatic embryos, they'll be mixed with sodium alginate and they'll be dropped into calcium nitride. Here we have seen, right, salts of calcium. So they'll be first of all they'll be mixed with sodium alginate and then they'll be dropped into calcium nitride, and surface complexion will begin immediately, and the drops, they'll be gelled completely within 30 minutes. It will take around 30 minutes for the formation of whole bead like structure, right? So gel complexion via dropping procedure. And the second method, that is the molding method. The isolated somatic embryos, they will be mixed in a temperature dependent gel, so temperature dependent gel, that is gel, right? And then they'll be placed in the microtiter plate and it will form gel as soon as it will cool down. So if you come across a question which is related to temperature related gels, so you'll be going for gel, right? So it's a company formulation, right? So this is the thing. Now if we are talking about artificial seeds, we have completed artificial seeds nicely, but now we need to test them and we need to do certain things that we have seen that it is tested. After that, if we want to change this artificial seed into embryo, what all things we need to do? So embryo into plant. This is our next question. So first is encapsulated embryos. They will be placed aseptically on simply agar medium with minimal nutrients, because embryo needs minimal nutrients. It doesn't need lots of what to say, inducing agents. It can go on a minimal medium. If we are saying embryo and it's a somatic embryo, so it should. So what we'll say encapsulated embryos are placed aseptically. That is very important. This word should not be skipped at all, because it is an artificial media. It can catch the bacterial and fungal contamination very quickly. So it is placed aseptically, though there are some antibiotics also added in this medium so that it cannot catch the infection very easily, but still we need to maintain aseptic condition aseptically on simply agar medium, simple agar medium. So simple carbohydrate medium is given. After that, uniform germination of somatic embryos and growth development of shoot and root system should occur. Root and shoot should develop. After that, production of true leaves. Production of true leaves should occur. So this is the way how embryo is changed into plants, right? But if you are saying this, there is one more thing which occurs in the normal rooting and shooting of our plant which grow in the soil, that is uh, hypsotrophy of hypocotyle. So that will be absence. That will be absent in this case. So absence of trophy of hypocotyle. This will be absent in this case. And a green plant with normal phenotype will be obtained. A green plant with normal phenotype will be obtained, right? So this is the thing. Now when we have come across all the things and we have seen everything about somatic embryo, we also need to know that how they are stored. So storage is a great thing when we talk about the somatic embryos or artificial seeds. So storage is a difficult issue. So this is the problem which makes it provides it great limitation. So when artificial seeds, they are stored at low temperature, the embryos, they show a characteristic drop in the conversion. They do not germinate immediately. All of them do not germinate. So drop in germination at low temperature, right? After that, this is the first problem, drop in the limited storage time of artificial seed is probably due to an anaerobic environment in the capsule. Now, when we are saying that it's a capsule, right? So it is not like a living seed. It is completely devoid of any kind of respiration. So it makes it anaerobic, quite anaerobic. A little change in the gel and it will be completely anaerobic. So anaerobic condition will make the seed again quite dormant, and the storage time is limited, or storage time is dropped. So storage time drops because of the capsule. So this problem of somatic embryos which are changed into artificial seeds, uh, they do not develop mental and they are arrested at a particular stage, right? Now we have seen that what are the limitations, and okay, one or two more limitations we would discuss. So that that are cost of labor, and material for the tissue culture process, and encapsulation and aseptic condition. Sorry for the disturbance. Encapsulation, then aseptic condition. These all are the limitations. Now, importance also one should discuss when we have seen it completely. So, what is the importance of artificial seeds? So, importance of artificial seeds. First, uh, the true seeds, they are produced in plant at the end of the reproductive phase, right, by the process of complex sexual reproduction and everything. So a plant will take long time to attain this reproductive phase. It does not attain it immediately, so we have to wait up till the end of the reproductive phase and then to get the seeds. But through artificial seeds we don't need to wait. We can just take any viable explant from that plant, and we can just go for development of as many seeds as we want to. So we would write over here that long time cut short. Long time cut short. Then plants, they bear flowers and they produce seeds at a particular season of year, isn't it? But the production of artificial seed, it is not at all season dependent. So importance of artificial seed, not at all season dependent. Season dependent. Now, occasionally what happens that work on some plants is delayed due to the presence of dormancy because of the environmental condition. So those all things can be overcome. So by growing artificial seeds, this period may be reduced, and using artificial seeds, the life cycle of a plant can be even shortened, right? So life cycle of plants shortened. After that, somatic embryogenesis has been observed in many great species till date, because there are many species which cannot propagate very easily, and they cannot be even transported to other place. So somatic embryos, it serves a very great source of development of many numbers of clonal propagates of these plants, right? Then artificial seeds will be applicable for large scale monocultures as well as mixed genotype plantation. So applicable for large scale monocultures. cultures, right? After that, it gives protection of meiotically unstable elite genotype. So protection of meiotically unstable elite genotype. After that, artificial seed coating has also the potential to hold and deliver beneficial adjuvants such as growth promoting thiobacteria, plant nutrients, and growth control agents, pesticides, and precise placement. So these are also the benefits. Then artificial seeds help to study the role of endosperm and seed coat formation. So that is very important, that artificial seed help to study role of endosperms and seed coat formation, right? So we have covered almost all the important points related to artificial seeds. We have discussed some artificial seeds, the definition, then we have seen the process of somatic embryogenesis, its definition, then what are the other synonyms which are used for the same, that is adventitious embryos, accessory embryos, embryo-like structure, embryos. These all things are given to the somatic embryos. After that, we have seen developmental stages of somatic embryos, that is 1 cell, 2 cell, 4 cell, globular stage, heart-shaped stage, torpedo stage. Then we have seen the types of somatic embryogenesis, then the ways of inducing somatic embryogenesis, then we have seen the application of somatic embryogenesis, then we have seen the artificial seeds, which was the actual topic. But before that, we should know somatic embryogenesis. Then we have seen the gel and the complexing agent, which is very important, the one which is temperature dependent gel, right, and everything. Then we have seen the two methods through which we actually drop the somatic embryo. So first you drop it into gel, and then you mix the complexing agent in it. Like if this is the alginate or the gel, then you have placed the somatic embryo in this, and then you mix the complexing agent, and you let it stay for 30 minutes, and then after 30 minutes you will see small artificial seeds, encapsulated somatic embryos. Then how to convert embryo into plant when we are talking about the encapsulated embryos. So there will be one important thing that will be missing. So that is the absence of trophy of the hypocotyl. This point will be missing. So remember this. This is very important, that which of these steps will be missing. So missing things are also important. This won't occur through artificial seed formation. Right, then we need to know how it is stored, whether storage is a problem or not, that thing. Then we have seen the importance of artificial seeds quite in detail. So like this we have covered the whole topic, and as we say in the starting and in the end, let's crack CSIR UGC NET together. And we are doing crash course for GATE 2021, GATE 2021. So be there, attend the classes. Every day I'll be taking class for GATE 2021 at 5 p.m. If you haven't subscribed our channel, just subscribe our channel, hit the bell icon for getting the notification. And if you're going for plus subscription, which can be very good for you to take the subscription now, because there is a major discount going on and soon the prices are going to increase. Do not forget to use my referral code, that is FP01, for getting an additional 10% discount. And even if you are going for the subscription through my code, you will have access towards all the videos and lectures done by any other educator as well. Not only my lectures, but all the educators who are teaching on the Unacademy, maybe on different channel. Any educator which is teaching in this category, that is CSIR UGC NET or CSIR JRF or GATE, these all things are like overlapping, so you will get access towards all the educators which are teaching. So do not forget to use my referral code, that is FP01. And if you really liked my video, give it a thumbs up. It really motivates educators. So give me a like if you really liked it. And lastly, a great thank you for your patient listening, and we'll be meeting tomorrow again at the same time, 5 o'clock, and we'll be doing some other topic in the category of GATE 2021. Any topic of the syllabus of GATE we'll be picking up. Mostly I'll be telling you about plant tissue cultures, and then methods and biotechnology, and all these type of things I'll be teaching you. So I think this is it for now. We'll be meeting again tomorrow. So do not forget to like the video. And one more thing, if you are interested in any other topic and you want me to teach you that specific topic, so just write down in the comment box or come tomorrow and write down in the live class, and I'll be very much willing to explain that topic to you. So this is it for now. We'll be meeting again tomorrow. It's a bye from